Good afternoon, everyone. My name is Daniel Aldea, and first of all, thanks for the introduction and for the opportunity to present my work. My name is Daniel Aldea. I'm a postdoc here in the laboratory head at Echever and Barlier at the Marseille Medical Genetic at the University Aix-Marseille here in the south of France. And today I'm very happy to present my work, and I'll talk about what we can learn about the biology of melanocytes by studying rare skin diseases. So next. So during human development, at around 2 months gestation, pre-melanocyte, which derive from neural crystal, they start to derive to reach most of the skin, but not just the skin. Next. We also know that melanocytes are found in other tissues, such as the central nervous system, eyes, a gland associated to the eyes, or even the heart. And during this presentation, I'll tell you about the main story that I'll tell you about is how mutations can affect the normal development of melanocytes. And in the last 2-3 minutes, I'll tell you about how the transcriptional program of melanocytes can vary across anatomical position or even within local environments. So in this case, in the environment of the skin. And this really has tremendous implication in our understanding of melanoma, which is a skin cancer, because we now know that oncogenic drivers, they will not behave in the similar way if they are located in a cell with an oncogenic driver. It will not behave the similar way if it's located in a body, or if it's located, for instance, in the extremities of our body, such as in the ventral area of the feet or hands. So next. So as I say, at around 2 months of gestation in human embryo, this pre-melanocyte, they start to migrate to reach most of the skin. And some of these melanocytes then might acquire a mutation, which can lead to an overproliferation of these melanocytes. Next. So in very simple terms, if this mutation occurs during gestation, it will manifest at birth as a skin disorder that is called congenital melanocytic nevus, which is characterized because of these large areas of the skin that are pigmented. Next. And it is commonly thought, on the contrary, that if this mutation occurs at birth or later on in life, it will manifest as what we know as acquired melanocytic nevus, which are commonly known as moles that are of small sizes. So despite the phenotypic differences, if we look at a molecular level, next, we can see that there are similarities. We can see that mutations that activate the MAP kinase pathway are found in both in congenital and in acquired melanocytic nevus. For instance, BRAF mutations are commonly found in congenital and acquired melanocytic nevi of small sizes, whereas NRAS mutations are commonly found in nevi of in congenital melanocytic nevi of medium and larger sizes. So these exact same mutations are also found in melanoma. However, we know that the malignant transformation of these melanocytes is rare, but it has been documented. So from that, one might ask, next, what other factors contribute to melanoma initiation in AMN and CMN? And what we know so far have been very well characterized that a hallmark of melanoma is a global DNA methylation changes. So we know, so the question is, we know that these mutations are not the only driver of melanoma, so there should be something else, is this global DNA methylation changes. So DNA methylation is basically, as you know, the addition of a methyl group to the cytosine in the ADN. So recent studies really have a focus on the melanoma initiation from acquired melanocytic nevi. However, there is a gap in our knowledge regarding melanoma initiation and progression from congenital melanocytic nevi. Next. So that's why in a laboratory, we decide to investigate that, and we decide to investigate the DNA methylation status of CMN samples at a genome-wide scale. So what we did was to collect skin samples from these patients. These patients, they harbor BRAF or NRAS mutations, and then to look to assess the methylation status of these samples, we use an array that is called a methylation epic, for which we can look at over 850,000 positions or cytosines genome-wide that are called CpG sites. And then, because we wanted to see which are molecular marker, we compare our data set with a relative similar study in terms of the mutations and in terms of the technology that they use. And also, in addition, this study contained normal skin. So next. So as I mentioned, these two types of nevi are very different at a phenotypical level. And this becomes really evident when we look at a histological level. So here you see a cross-section of immunohistochemistry for which we stain SOX10. SOX10 is a well-known marker of melanocyte, and P63, this is a marker of the epidermis. And you can see that in the CMA samples, you can see that melanocytes go really deep into the dermis from the epidermis at the top, go really deep into the dermis. And this really contrasts with what we observe in acquired melanocytic nevi, in which you can see that melanocytes are most at the surface of the skin. We can observe melanocytes in the dermis, but very much less than compared to CNN. So these same phenotypic differences, it can be uncovered, it can be revealed if we use our methylome data. And this is what we did. Next. And this is what we did. We used the same data, the same methylomic data to reveal this difference. So here in this plot, you can see each column represent a sample, represent a patient. And within this column, you can see the cellular composition in terms of melanocyte, keratinocyte, or fibroblasts. So you can clearly observe that for the case overall, that's for the case of CMA, we observe more melanocytes in orange compared to AMN or compared to a control skin. You can also observe that there is a huge variability even within CMA samples in terms of the number of melanocytes or the proportion of melanocytes that these samples they have. So given that, what we did was to correct this data, to consider, to take into account these differences in terms of the number of, in terms of the cellular proportion. So once we did this using bioinformatic tools, then we were able to really look for differences, molecular differences between these different types of samples. So next. So when we look at a global level, we observe that CNN, or congenital melanocytic nevi, are globally less methylated than AMN, but not different to the skin. So you can observe here, each dot represents a sample that is the average methylation across over 700,000 positions. And you can clearly observe what I just said. CMN is less methylated compared to AMN, but is similar to the skin. So next. We then want to look at which are the differentially methylated positions between CMN versus AMN. And we observe that the number of DMP are most of them, they were located in intergenic or promoter regions, which are regulatory regions. And then when we look at in detail, next, we observed that the top DMP in the promoter region, what caught our attention was that this gene that is called spectrin beta, for which we observe many of these CpGs, located at the promoter region of this gene. And that means that they were hypomethylated compared in CMA compared to AMN. So next. So this is the genomic landscape of spectrin beta. And you can observe what I just mentioned, that many of these CPGs, they were located in an alternative promoter of this gene. Next. And we observe that for each of these positions, we observe that the methylation was less in CMN compared to AMN, but not to the skin. So it is very well known that hypomethylation of a promoter is correlate with an activation of a gene. So that's why we thought that let's go to see where this gene is expressed in CMA samples. Let's see if there is any difference with AMN samples. Next. So to do that, that's why we perform an immunofluorescence. So here you see a column. So here you see the CMA samples and the AMN samples. And you can observe that spectrin beta is really nicely expressed in the epidermis, about the line in green. And this was already described, so we validate this with immunofluorescence. But I really want to draw your attention to the dermal area in which we can see expression of SOX10, which is a marker, of melanocytes. And you can see that we have a really strong expression of spectrin beta in this area, compared to the few dermal cells that we found for the case of AMN, in which spectrin beta is not done as much as expressed as in the case of CMA. So these together really put spectrin beta as a potential marker to distinguish between CMA and AAM, and so only by using the methylome data that you can get and that are easily to get in many hospital in setup of hospital. Next. And now I just want to, I would love to continue that story, but I just want to switch gear a little bit and tell you about what are the effort or what we are doing for to understand or to uncover the transcriptional program differences of melanocytes between the different anatomical positions and within the local environment. As I just mentioned, this really has a tremendous implication for our understanding of melanoma, because really now we know that cells that harbor an oncogenic driver, it will behave, or it will not initiate melanoma if it's located, for instance, in the body, but it will initiate melanoma if it's located on acral area that are there, this area of the palm and the soles in the feet, because there is a different environment, a different cellular niche in this area. So next. So to do that, what we are using, we are taking advantage of, we turn to a mice. And what most of the biology that we know about melanocyte is coming from a study that has been done in the back skin, where you can find melanocytes. And really little attention has been put on the melanocytes that are residing in the limbs area. We can use enzymatic dissociation to separate dermis and dermis. In both, we can find melanocytes. And also, we can use this animal model to study the melanocytes that are associated to glands that are in close proximity to the eyes. For instance, the Harderian glands. You can see this is full of melanocytes. As well, you can find melanocytes in the brain, in the meninges. So really, this question is understand what are the difference between these different populations of melanocytes that are homologous to the humans, to then understand what are these transcriptional differences, and why some oncogenic driver, it will initiate melanoma in some tissues, but not in others. So stay tuned for this story. Hopefully very soon we will tell you more about that. And finally, next. I just want to tell you that what the effort that we are doing to understand about the within the tissue, about the local environment. So to address this question, we turn to a spatial transcriptomic. That is a technology that allows us to observe or to look for differential expression without losing spatial resolution. So remember at the beginning, I mentioned that in CMA, in congenital melanocytic nevus, you have melanocytes that go really deep into the dermis. And one question that really fascinates me is try to understand if the melanocytes that are in close proximity to the epidermis, are they the same to those that are like deep into the dermis? So to address this question, as I mentioned, we are using this technology. You can see that actually, when we cluster this melanocyte, we observe already transcriptomic differences. You can observe here MIT, MITF, that is a master regulator of melanocyte. It's very well expressed in all the melanocytes in this tissue. And we already started observing some differences at the transcriptomic level. For instance, OCA2 is a gene that is expressed much more on the melanocytes that are in close proximity to the epidermis, but not much to those that are deep. We find this another example, like TA1 is a gene that is expressed much in the deep layer in the dermis, but not in the other part. So like this is a work in progress. It's something that we are continuing in the lab. And with this, I just, next and final. I just want to say thank you to everyone involved in this work, to the team, especially Heather, Elise, and Matthias, and funding sources, and happy to answer any question.
Great, thank you very much, Daniel. That was great. So again, if you have any questions, please do put those into the Q&A box. And while we're waiting for that, I was just wondering whether you could tell us a little bit more about what's understood about what causes those changes in methylation status. And similarly, are your CMN cells more similar in their methylation status to then melanoma cells that are coming out of that? And so are the melanomas more likely to arise from CMN, or is there a link between that, those two conditions?
Could you repeat the first question?
Yeah, so my question was, what's understood about what's the cause between those, what's the sort of cause of those differences in methylation status between AMN and CMN?
Do you mean like if there's any difference like between them?
Well, so as in you've got the same driver mutations, but in one case you've got hypermethylation and in the other case you haven't. So given that those cells are genetically similar, what's leading to those changes in methylation status?
Yeah, that is something that we are investigating right now. This is you can see that also the spectrin beta is expressed in both. It is just a marker that is more expressed in CMA. We actually don't know exactly what is the differences between both. What I can tell a little bit is something about the mutation, because if you see the data, in AMM we find more BRAF mutation compared to CMA in my data. So maybe there is a difference between the mutation that is driving there and causing like the difference on methylation.
Okay, thank you. And then we've just got time for one more quick question that's in the Q&A. So this is Ruth Williams, says, very interesting work. Have you or anyone else done single-cell RNA-seq analysis of melanocytes from different tissues? And what other genes are differentially expressed, like SOX10?
Yeah, there are some work done in single-cell RNA-seq analysis of melanocytes, not from different areas in the body, though. Most of the work that have been done is in the skin, in back skin, or in human in the part of the body. Whether other genes are differentially expressed like SOX10. Yeah, as I said, we don't know much about that because there is no much single-cell RNA-seq studies.